A very good afternoon to all. We have our next session chair, resource person, Dr. K. Anand, DTAC and Pre-Cam Senior Associate, and he is a research scientist, NRF DSL Fellow at Department of Chemical Pathology, School of Pathology, University of Free State, Bloemfontein, South Africa. It's my privilege to brief his profile. Dr. Anand has expertise in organic chemistry, medicinal biochemistry, integrative medicine, nanobiotechnology, and drug discovery. He has received doctoral degree in organic chemistry in the Department of Chemistry, Durban University of Technology, in collaboration with Department of Medicinal Biochemistry, University of KwaZulu-Natal in 2014. He has published many scientific articles in international peer-reviewed journal and has authored many chapters as well as review article. He has recognized for his contribution and received various awards from national and international organizations. He has been awarded Best Doctoral Research Award for 2016 and and nanotoxicology and chemical pathology. Currently, he received a prestigious Innovation Research Fellowship from National Chemical Pathology, School of Pathology, Faculty of Science and National Health Laboratory Services, University of Free State, Bloemfontein, South Africa. And it's really, he's having a brief profile, but in short, I can compile. And I welcome once again Dr. K. Anand.
Thank you. Thank you. So now I can start now.
Yes, sure.
Okay. Now I'll share my. Okay, now you can see my slide now.
Now, perfect. Welcome, Dr.
Yeah.
Anand. Yes, it's visible and
Okay.
your audio is also
Okay, everything perfect now.
perfect. Start.
To reach
Speak
India.
a little bit loud.
Okay. This is Dr. Anand from South Africa. I'm basically from India. I came to South Africa 2009 to do my PhD. Then I started my research career with the Durban University of Technology in South Africa. I did my PhD there. Then I got postdoc opportunity in University of KwaZulu-Natal. I started my research career there. 3 years I worked with the medicinal biochemistry laboratory with Professor Anil Chatakon. Then I again molecular biology research methodology from that lab. Then recently, last year, I joined with the University of Free State, which I'm showing my first slide, as a scientist with the National Healthy Laboratory Service. This is my University of Free State, one of the famous university in South Africa. The city is called Bloemfontein. Bloemfontein is the best place for like education. The city is a lot of historical background behind the city, mostly from German people settled in South Africa. So the University of the Free State made by who came from German, they developed this university. Okay, so now currently I'm working in this university as a scientist, Department of Chemical Pathology. Okay, so today I'm going to interact with the topic exosomes. So which I started 2 years before this research work through my postdoc laboratory. It's very interesting research topic. The young researcher who would like to start research career with the drug discovery, diagnostics, disease biomarkers, so they can start up with the exosome research field, because the exosome lot of hidden things is there. So still research going on, so people can start with this topic. Okay, so today I'm going to talk about exosome, the next nano thing. Okay, so here why we mentioning it's like a nano. Bios is exosome made from our human body. I will explain clearly one by one. Okay. Okay. Now I'm going my next slide. Okay. I like to explain about briefly what I did before, what I'm doing now. Okay. When I started my research career with this PhD laboratory, I started with the synthetic chemistry, synthesizing new molecule from small molecules. So you can see my slide here. You can see some of the chemical structure that's called nitrogen heterocyclic, like ellipticin, tryptosin. These are a carbosol family small molecules. These are very good DNA intercalator, biomolecular intercalator, protein intercalator. So I started my research career with synthesizing novel drug molecule. Then side by side, I did some work with the plant pure compound isolation, then testing with the different disease. Okay, so for example, South Africa indigenous plant, I am collecting from plant, we isolating some compound, then when testing with the different disease like a cancer and HIV. So these are work I did before. Okay, then as a chemist, then I start think about when nanotechnology. Then as a chemist, as an organic chemist, how move to synthetic to nano technology. So during my PhD, I synthesized a lot of compound. So that time I designed some project to self-assembly nanoparticle. Okay. So for example, what is the self-assembly here? There are a lot of different techniques in the self-assembly nanoparticle synthesis. But as an organic chemist, in my lab atmosphere, I designed some methodology. For example, I take water-soluble compound, I take water-insoluble compound. Both compound is biologically active. Okay, but it's active. The biologically active means both is different properties there, but I like to create synergistic effect between the two molecules to convert into nanoparticle. Okay. For example, I took aminocarbosol. You can see the corner of left side corner of small molecule aminocarbosol. Then the aminocarbosol very good for DNA intercalation. Okay. So another one molecule you can see lipoic acid is a very good antioxidant from natural product. So what I did, I react together, then I create a amide bond, then I convert into one molecule. So two molecule I convert into one molecule. As like a combine together. Okay. So what happened here? So whenever you react with water soluble and water insoluble, then you create a solvent emulsion. It converted to nanoparticle. Okay. So so this is a self-assembly nanoparticle from synthesis molecule. Okay. Then we testing with the cancer cell line. We did characterization. So this is one of my PhD work, from pure synthetic to nanoparticle synthesis. I moved this. Then I started reading about some how to synthesis nanoparticle chemically, then how to use that nanoparticle as a drug. So these are some picture I'm showing. Um, some plant material, how to use plant extract, how to use as a nanoparticle synthesis. Okay, after synthesis, how to use that nanoparticle, plant derived nanoparticle to drug delivery or like a sensing. So these are this project I did some of the chapters, my PhD chapters. Okay. Okay. Now, how the plant compound interacting with the surface. Plant compound naturally antioxidant. So antioxidant have properties to reduce metal salt. So it will reduce metal salt to metal nanoparticle. Okay. So these are metal plus plant antioxidant combining together. It give very good biological active. Okay. So we have published some article with this research field. We got some good article. Okay. Then what I did, okay. Then the slide showing same thing, Vito nanoparticle, same plant extract, we taking plant extract, then we mix with the metal salt, we try to make metal nanoparticle capped with the plant compound. So previously what we did, we, we used to take directly plant extract, then we synthesis molecule. Then what I decided, we try to pure that plant extract, try to take pure compound, then I react with metal salt, then convert into nanoparticle. This kind of project I did. After sometime when you're sending some, when you're doing some work, we publishing some paper, some reviewer, editor giving some advice, some command. Based on that command and advice, we have to move further to develop our project. So this is the project we develop based on reviewer and editor command. So you can see we publish some quite nice, good journal like Wiley and Elsevier. So we got some good journal from this kind of work. Okay. Okay. Now, okay. So previously I explained about what is my research background, synthetic background, nanoparticle synthesis, and plant derived nanoparticle synthesis. So these are the work I did before. Okay. So I gain knowledge, synthetic background, nanotechnology background, as well as biological background. Okay. Then I decided to read about a lot of article to develop my project. Then I found some interesting topic related to nanotechnology. That's called exosome. Okay. Now you can see this video. I like to share some one video. So where is generating and then how communicating with one cell to another cell. So I'm showing some video. You can see that video. Okay, you can see here one cell, another cell, they're connecting through nanotubular, like small fiber like that. You can see cell image in a microscopy. You can see here the particle, some particle migrating to one cell to another cell. So what is that particle? So that particle, what you doing there? So that particles maybe some proteins or some virus and something like that. So we need to find out what is the particle here. So researcher tried to proving these particles also called as a exosome. So this involving cell-to-cell communication, taking a message to one cell to another cells. So this is very important role here. Okay. So that video is very interesting. Now you can see some picture here. The cell in between two cells, some particle migrating to one cell to another cell. So that particle is what is this? That is particle is also called exosome. Okay. Okay. Now, what is the exosome? What is the exosome containing made with what is this? And then is containing what is inside. Okay. So the exosome made with the protein. The inside the exosome, you can see here lot of biological molecule like protein and DNA and microRNA. So these are related to biomarkers. So something inside this exosome containing something. So it's taking parent cell to donor cell. So that means it's a simple concept. Something taking the message to one cell to another cell. So if we trace that exosome, we can find out what is it transferring to one cell to another cell. So then means something communication between two cells. That means is maybe is spreading the disease or spreading the some good things or bad things. Okay, we don't know, but we need to find out what is taking to one to another. Okay, so these are the exosome. This size is like a nanometer level. It's containing DNA and microRNA and protein. So the lot of biomarkers is there. So people are working with this exosome, try to find out what is there. Okay. Also, one more thing, very important. Every exosome, every body. I will explain about other what is where we getting exosome, how is forming. I'm explain next slide. Okay. So every organ have different type of exosome. For example, cancer. For example, lungs organ have lungs based exosome. So kidney based exosome. So the different organ generating different type of exosome. That means, for example, for kidney related disease, we can find out biomarker from urine. Some blood, so blood related disease we can find out from that blood based exosome. So different exosome from different part we can identify. Okay. Okay, now I'm just telling you, just sharing about exosome characteristics. Okay, what is the exosome? How we can find out from other? For example, cell have different particle. Like a cell have different, maybe like for example, nucleus, some cytosols. But exosome have different properties from this cell surface. Okay, so exosome is size is 30 nanometer to 100 nanometer, and is mostly the exosome finding from body fluid, like blood and urine and saliva and milk, like so whatever our body fluid is there, we can find out the exosome there. Then it export by all the aeropatic cell types. Okay, then exosome properties, here is the main point here. Exosome properties reflect the donor cell. Okay, so the properties from donor cell means like parent cell. Okay, one, for example, every cell have some communication. So the parent cell is taking the genetic matter to the other cell. So exosome property referred to the donor cell. Okay, it produces extremely abundant amount. Okay, these are the exosomes of biogenous from donor to target cell. I saw some picture. You can see the donor cell delivering exosome to receptor cell. The exosome containing DNA, protein. They have some surface protein like that's called CD9, CD82, and you can see the inside DNA is there. Some enzyme is there. Biomarker for disease. Okay, because okay, now you can see the application by using the exosome, how they are incorporating. Using this exosome different field, like first one biomarkers, more then targets, and then delivery vehicle. Okay, so now you can see the biomarkers part. So for example, one patient have kidney disorder. Okay, maybe we can take blood. We doing some test in a laboratory. We can find out this patient have this is kidney disorder or metabolic syndrome. Something we can our report pathologic researcher give report based on that test. But still, lot of disease, like for example cancer. So even though we try to find out cancer biomarkers, still hidden thing is there. How to prevent the cancer before it's come. So we need to some biomarkers there. Our human body releasing some biomarkers before the cancers come. Okay, so we need to find out the exact biomarkers. So the exosome may be carrying the biomarkers before the disease come. So if we catch up that, if we track that exosome, if we study the exosome, we can find out in one children, one child in future, maybe it getting Alzheimer, maybe the child will get cancer. So we can find out beginning of the stage before it come, because the exosome is I'm saying, I previously I discussed exosome carrying a lot of biomarkers, say hidden things. Okay, so there's a main role is it carrying a lot of biomarkers. Okay, so this part applying to diagnostic field. Okay, then come to target. Okay, these are the target, like amyloid and the tocoproteins. So the target means like a proteins. Okay, it have some proteins. Mostly drug designing and drug discovery people try to find out target, like by using and some bioinformatics. Okay, so far we designing a drug based on cell express proteins. Okay, for example, cancer cell express some protein or a kinase protein or some K space, whatever. Cell based target only we designing drug molecule. But so far we didn't design drug molecule based on exosome expressed biomolecule, like exosomes expressing some protein, exosome express some biomolecule. If we designing our drug molecule based on exosome expressed biomolecule, maybe we can control the disease, like how we designing drug molecule cancer express protein. Okay, same thing. Okay, why here, why we try to target exosome proteins or exosome biomolecule? The reason is the exosome, I told you, the exosome taking genetic message to one cell to another cell. For example, the cancer cells, for example, cancer starting point, one cell to another cell, uh, taking one cell the cancer message to spreading to another cell. Okay, then means this is the main role spreading that cancer disease. Okay, so if we target the exosome, if we inhibit the exosome protein or exosome protein function, maybe we can control exosome transportation. Okay, so we can stop, like for example, like one, for example, we one bus going to one place to another place, we stopping that bus, we don't want to go that place. Okay, so same thing. Okay, so that is a target, like drug molecule designing that field. Okay, drug designing, target designing, identification, that field covering that area. Okay, the another one area is drug delivery vehicle. Not only drug delivery vehicle, even RNA delivery or drug molecule delivery or protein delivery, because the exosome is like a spherical. Okay, so it have some receptor. Okay, um, surface protein. Okay, so the exosome know which cell to which cell communicate. Okay, for example, cancer cell to the normal cell, um, it transferring to one cell to another cell. Okay, so now people, some of the researcher, they isolating exosome, they using the exosome as a drug delivery agent, like a libosome, maybe like nano delivery. Okay, they isolating exosome, they loading to drug molecule or RNA therapy, RNA molecule, they're delivering to um, like a target molecule, target site, like a target cells. Okay, so the using is a delivery vehicle. So from this slide you can see exosome we can use as a biomarkers. Exosome we can design as a protein targets or biomolecule target drug designing area. Also the exosome we can use as a drug delivery vehicle. Okay, so three field covering. Okay, okay, now diagnosis. Okay, why we want to diagnose this exosome? Okay, see, now a lot of diagnosis method is there, protein diagnosis, a lot of diagnosis method is there. By using the diagnostic method, we giving report disease, patient have this disease, the disease condition, the physiology of the disease, pathology of the disease. So far we diagnosing with the blood and urine, or these are some pathology lab helping for diagnosing. But in future, exosome diagnosis method is very important. Okay, very important as well as very useful. Okay, because the exosome is a carrying the disease biomarkers. So if we capture the exosome, if we analyze the exosome, we can find out the exact disease biomarker. Cancer again, people used to go to see GP, general practitioner, then they will give some, then they will take blood or urine, they go for a diagnostic lab, the testing, then they will say, I got cancer, you got cancer, you got this disease or that disease. So they're giving a report like that. But exosome diagnosis, not like that. Exosome diagnosis, for example, one 30 years old young man, after 60 years, 60 years, maybe he will get, we don't know, he will get Alzheimer, he will get some neuro disorder. People can, the researcher can find out before it will come out his body. It would come out, it will come in his, like before predicting like that. Maybe future, this person will get this disease. So this exosome is a very important role in future. Okay, so now people are, you can see people are starting the exosome diagnosis research field. Okay, so like is beating the traditional diagnostic methodology. Okay. Okay, now this slide you can see exosome target delivery. Okay, so I told you before, we can use exosome as a drug delivery because the exosome surface has some protein. That protein know where can it find, where can it attach, because it's already made up with, naturally made up with some protein. Okay, so we loading our drug molecule in the exosome, we targeting the disease cells like a cancer cell or whatever. Okay, so for example, this slide will say, you can see curcumin. So like they're using this product is there already. So the curcumin, the loading with the exosome, the targeting to disease cells. Okay, so they're using as a delivering agent. Okay, now you can see future therapy, immunotherapy, by using exosome, how they are going to treat patient, like immunotherapy method. Okay, here you can see the slide, the patient is center. Okay, for example, the patient have cancer. Okay, so we need to find out the patient genetic, what genetic. So we need to find out like exactly the patient what he having. Okay, so the exosome give proper identification based on the patient. So now you can see, first we isolating exosome from the patient, we try to find out exact biomarker for that patient disease. Then we designing the drug molecule for that patient. Then we loading drug to the same exosome. We delivering to same the patient. So it means, how can say, our body itself have medicine, or like we do no need to bring medicine from outside. Our body, our every human body, every patient has self immuno responsible. Okay, so it's going to the exosome immunotherapy. So there's a research still is going on. So you can see this. Okay, now the I explain about what is the exosome, what is the use for exosome, what is the application, and what is the property of exosome. I explained the old slide. Still, I like to share exosome. It's a different type of exosome is there, like a urine-based exosome, blood-based, like a serum-based exosome, plasma-based exosome, and saliva-based exosome. There are different type of exosomes there, based on different type of organs. Exosome is there. For example, lungs fluid have one type of exosome. Uh, our final central nervous final fluid has different type of exosome. So different part uh, will different part give different type of exosome. That different type of exosome give different type of biomarkers based on the human the patient condition. Okay, now I'm going to enter now my work so far, what I did in my lab with the exosome. Okay, there are different type of project we doing. One is exosome sensor. Okay, I'm not focus on still diagnostic in exosome. I'm focusing sensing the exosome. Okay, as yes, this is a basic research how to sensing the exosome with the different techniques. Okay, because we need to find see people are doing research with the isolation method, characterization, and developing functional modification. The three type of field they're doing research. So the researcher try to find out new isolation method, new sensing method. So because like for example, when isolation kit we using our laboratory. Same thing. This research how to isolate exosome, how to identify. So this type of exosome, this type of work we doing. Okay. The another work is we try to incubate exosome. Like we designing the drug molecule to control the exosome. Okay, based on the drug molecule, we incubating that exosome protein or activating the exosome protein. We controlling the exosome. So that is also we doing some work. Okay, future we going to do diagnostic. Okay, now this one work I'm going to discuss cell derivative vehicle exosome gold nanoparticle interaction. Okay, this hormone like effect and self assembly and smartphone sensing. Okay, these are the basic research in our lab. So gold, why I'm choosing the gold nanoparticle here? See, people used to sense chemical sensor and the flow biosensor is there. So the future nano sensor is there because it's the same thing. Like a chemical sensor, they're using organic dyes molecule like a fluorescent dyes to sensing that some biomolecules. So they're reacting with the protein molecule or reacting with some biomolecule, the chemical losing that properties. So based on the losing the properties, we detecting some method like UV absorption or UV fluorescence, the instrument like whatever microscopy instrument, bioimaging. So based on the chemical losing the properties, so we identifying so we using as here the chemical is a PROF, molecular sensing device. Okay, so same thing nowadays the nanoparticles involving instead of the chemical, the nanoparticle involving that is sensing on PROF, like small sensor like that. Especially the gold nanoparticle is a very important role here because the gold nanoparticle have very good surface resonance properties. Okay, that means it have some UV absorption emission property. So people start using the gold nanoparticle as a sensing device, sensing probe, small like dice like that. Okay, so we use this method like colorimetric detection by using gold nanoparticle. Okay, because colorimetric is a very good method to detect. For example, ELISA, like same like ELISA test, we the ELISA kits microscopy by we reading that UV value, and then we plotting that curve. Okay, same thing. We using the colorimetric detection by using golden nanoparticle. Okay, so first step we synthesis the gold nanoparticle with capped with some biomolecules. The biomolecules, small organic molecule, which can interact with the exosome. So before you designing that molecule, we need to work with that the capping, the golden nanopod capping molecule. Uh, we need to theoretically we need to work with this molecule bind with the exosome protein or incubating or activating. So we need to see clearly. Then only we take that compound, then we kept with the gold nanoparticle, then we react with the exosome. This is the concept we apply. Gold nanoparticle just for color changing, like colorimetric detection, like what I say, surface plasma property changing, like is naturally you have, for example, you can see here, it's naturally gives a like a red color. So when you changing, when you're adding some external like external protein or external substance, when you're adding that gold nanoparticle, it will react with that external substance, it change the color. Based on change the color, we can detect that UV value, UV value. Then we can get some data. Based on the data, we can work out. Okay. Okay. This you can see here the graph will abstract of my whole work. So you can see the left side, I'm synthesizing gold nanoparticle by using chemical method. Then I'm taking the methylene, so one of the small molecule, which is easily interact with the exosome surface protein. Okay. So before I react, I kept with the gold nanoparticle. I separately I work with the methylene with the exosome surface protein. Is how is interacting interaction properties. So we're working theoretically with the pure molecule. Then the molecules have some property to interact with the exosome protein. Then we capping with the gold surface. Then we using that gold cap and that molecule capping capped gold nanoparticle as a sensor device like okay. So you can see my whole work here. First, I'm synthesizing gold nanoparticle. Then I'm capping with my small molecule that's called a methylene. Then I'm reacting with the self-surface protein. Then I'm doing some theoretical studies, and then I'm doing experimental studies. I'm proving my gold nanoparticle is interact with the exosome surface. Okay. Okay, these are experimental section. First method is synthesizing methylene gapped gold nanoparticle, then reacting with the exosome different concentration, then I'm characterizing this combination, then application. Okay, so these are my experimental section. These are a lot of there are a lot of techniques is there, but I'm just showing small like UV. We can detect that binding. Binding properties, the SEM and the TEM, you can see the nanoparticle morphology. Okay, then I'm explaining theoretically how is the reduction, how is capping with the golden nanoparticle. I'm explaining theoretically because as a chemist, I like to explain the theoreticals. We cannot say simply is capping. We need to explain chemical point of view. Is getting oxidation or reduction something. Okay, so these are the slide explained about all. Now you can see the HR term, the binding site, how the golden nanoparticle interactive with the exosome surface. Okay, you can see left side, you can see pure exosome with the meglunamine. Okay, the right side you can see exosome with the golden nanoparticle. You can see the dark, the particle is a golden nanoparticle. The another one like a big particle is that is the exosome. Okay, so there's a two picture is there. Golden nanoparticle, you can see theoretically we study there are two binding sites, but when attaching with the golden nanoparticle, it's creating 5 binding sites. So we improving that binding site because of we need to bind bind with our protein like strong. Okay, so these are experimental we did. Okay. Okay, now you can see this Warman like effect. Okay, here, okay, we designing the molecule, we doing experimental, we characterizing, we proving our result. These are some hypothesis always is coming our project. Okay, this is the hypothesis. We need to prove that hypothesis how that exosome interacting with the gold surface. We need to prove through some some theory. Okay, so this theory is a Warman effect theory is a very good theory for surface plus properties. For example, you can see here. Um, you can see here on surface some some molecule sitting in the surface. When other molecule coming the top of the surface, that already the molecule sitting is moving towards to next. Okay, you can see here the blue color spherical is here some molecule. It's a charged molecule. Okay, so other the red color molecule coming from top, it's coming from, like for example, exosome. The blue color molecule considered as a cold nanoparticle. So is exchanging like the surface. Okay, that means the exchange surface means the surface properties changing. Okay, so these are the Warman effect proving this concept. So we need to prove this Warman effect is by using some experimental technique, some instrumentation. So I use zeta potential. Okay, normally people used to do zeta potential for just detecting size and